Chromatin Immunoprecipitation Sequencing, or ChIP-Seq, is a technique used to identify where specific proteins bind to DNA across the genome. It combines chromatin immunoprecipitation with high-throughput sequencing. So how does it work? In the standard or cross-linked ChIP-Seq workflow, proteins are first cross-linked to DNA in living cells, typically using formaldehyde. This freezes protein-DNA interactions at a specific moment in time. The cross-linking reaction is then usually quenched with glycine to stop further fixation. The cells are lysed and the chromatin is fragmented into smaller pieces, usually by sonication or in some protocols, enzymatic digestion. Although most ChIP-Seq protocols use formaldehyde cross-linking, an alternative approach known as native ChIP and ChIP omits this step. It is particularly well suited for studying histones and their modifications, since histones are already tightly associated with DNA. In this method, nuclei are isolated from gently lysed cells and the chromatin is digested, typically with micrococcal nuclease, to release individual or small groups of nucleosomes. Next, an antibody specific to the protein of interest is added to bind the protein DNA complexes. Magnetic beads coated with protein A or protein G are then introduced. These proteins bind strongly to regions of the antibodies, allowing the antibody-protein DNA complexes to attach securely to the beads. When the sample is placed in a magnetic field, the beads and the bound complexes are pulled to the side of the tube. This allows unbound DNA, proteins, and other cellular components to be washed away. The result is a highly enriched collection of DNA fragments specifically associated with the target protein, representing the genomic regions where the protein was interacting with DNA. After washing, the enriched chromatin is eluted from the beads, typically using heat and an elution buffer containing detergent. In cross-linked ChIP, the formaldehyde cross-links are then reversed by prolonged heating, usually at around 65 degrees Celsius in the presence of high salt. Proteinase K is added to digest the proteins, including the target protein and any residual antibody. In native ChIP, because no cross-linking was used, the cross-link reversal step is unnecessary, although protein digestion and DNA purification are still required. Once proteins have been removed, the DNA is purified, commonly using spin columns or magnetic bead-based cleanup to remove residual proteins, salts, enzymes, and other contaminants. The recovered DNA fragments then undergo library preparation. This includes end repair, addition of a single adenine base to the 3' ends known as A-tailing, and ligation of sequencing adapters. These adapters enable PCR amplification and sequencing. The resulting reads are aligned to a reference genome. Computational peak calling algorithms then identify regions of significant read enrichment corresponding to likely protein binding sites. If you'd like to test your understanding of what we've talked about so far, pause the video and try the interactive quiz question. Now that we've covered the core workflow, let's look at some important factors that influence the quality and interpretation of ChIP-Seq data. A key point to remember is that protein-DNA interactions are dynamic and highly condition dependent. If a transcription factor is inactive at the time of sampling, little or no binding signal may be detected. Careful experimental design, such as applying specific stimuli, performing time course studies, and including biological replicates, helps capture these transient interactions. Appropriate controls are equally essential. Input DNA controls account for biases in chromatin accessibility and fragmentation, while immunoglobulin G controls help identify non-specific antibody binding and background enrichment. Together, these controls increase confidence in the identification of true binding sites. Pause the video to try another question, this time on ChIP-Seq experimental design. Let's now talk about single-cell ChIP-Seq. While traditional ChIP-Seq analyzes bulk populations of cells, emerging single-cell methods allow protein-DNA interactions to be studied at cellular resolution, revealing differences between individual cells. Single-cell ChIP-Seq follows the same fundamental principles as bulk ChIP-Seq, but it is adapted for extremely small amounts of starting material. Individual cells are first isolated, often using microfluidic or droplet-based systems. Protein-DNA interactions are then preserved, and chromatin is fragmented within each cell or nucleus. To improve efficiency, many modern protocols introduce unique molecular barcodes early in the workflow, often before pooling samples. This allows chromatin from many individual cells to be combined and processed together while preserving the identity of each cell during sequencing analysis. Following immunoprecipitation with an antibody specific to the protein or histone modification of interest, the enriched DNA is recovered, crosslinks are reversed if crosslinking was used, and sequencing libraries are prepared. A major challenge in single-cell ChIP-Seq is data sparsity. Because each cell contributes only a small number of DNA fragments, it can be difficult to distinguish true biological absence of a signal from technical dropout. Pause here and see if you can recall what was mentioned about single-cell ChIP-Seq. While ChIP-Seq is a powerful tool, it becomes even more powerful when interpreted alongside complementary genomic approaches. Transcriptomic methods, such as RNA-seq, measure gene expression, while chromatin accessibility assays, such as ATAC-seq, identify regions of open chromatin. Together, these approaches provide a more complete picture of gene regulation. Now try one last question. I hope you found this video useful. If you did, please support the channel by liking the video and subscribing. Thank you for watching.